Hi, thanks so much for tuning in. My name is Breno Silva. I'm a chemistry major and environmental science minor at Suffolk University. I'm currently working on a research project with Dr. Denyce Wicht from the Department of Chemistry and Biochemistry. And this project is sponsored by the McNair Scholars Program as well as the National Science Foundation. The title of the project is Determining Kinetic Parameters for the SfnG Catalyzed Conversion of DMSO2, which is also called dimethyl into methanesulfinate. So a little bit of context on this project. It is very focused on sulfur and the global sulfur cycle, which is basically the way that the element sulfur is transformed and moved around the planet through different compounds and processes. And in terrestrial environments, this cycle is heavily driven by bacteria and other microorganisms. And the main way in which they use sulfur is through inorganic sulfur compounds. So this one here, this last one is called sulfite. This is a really common one that they use. And they use it mainly for energy and nutrients and such. However, an issue with this dependence is that not every type of soil or type of land is rich in inorganic sulfur compounds. So then the scenario that we're looking at for this study is how this bacteria assimilates sulfur from organic sulfur compounds. And the difference between organic and inorganic compounds is the presence of carbon atoms. So whenever carbon is seen, such as here in a compound, it is an organic compound. And an issue with this is that this carbon sulfur bond here that you see is particularly strong. So it's much harder for the bacteria and these other microorganisms to break those compounds up to get the energy that they need. So my mentor has been looking at the scenario for a few years, and she's proposed this mechanism for this assimilation of sulfur. So this first one that we see here on the top is her proposed mechanism. It is a 3-step mechanism, as you see here, and each step uses a different enzyme to catalyze a specific type of reaction that ultimately is aiming at converting organic and inorganic sulfur compound. In our case here is this DMSO2 molecule, this dimethylsulfone, and ultimately this is going to be converted into inorganic sulfite. You see there are no carbons here. So this is now inorganic and much easier for the bacteria to use that compound for what it needs it for. And I specifically will be looking into this first step. So this here is what you see in the title, the conversion of DMSO2 into methanesulfinate catalyzed by the protein SfnG. And this down here is the expanded version. So these are all of the reagents that will be required, my catalyst, my enzyme, and then the product. And then my main product is methanesulfinate here, which will then go on to react until it is turned into sulfite. The protein will be obtained from the bacteria Pseudomonas fluorescens. This is one of the bacteria that has the genetic material required to produce these types of protein to undergo this reaction. And this is basically what we're modeling in the lab. This process that does happen in nature once DMSO2 is deposited in soil. And the big question that I will be answering is, given the required condition, how fast does this reaction happen, which is a different way of saying I'll be determining the kinetic parameters for the reaction, which are going to tell me how viable this process will be in nature. And some things to note about this study, biochemical processes such as the one that I'm looking at, they greatly impact agriculture. So there is a great deal of agricultural relevance to it. And a big problem with this topic is that there is very limited knowledge on this consumption of DMSO2 by bacteria, which is another way of saying that not that many studies have been conducted looking specifically at this process. There was one study that I found that shows that there is some bioaccumulation of dimethylsulfone in plants once it is deposited in soil. And therefore, this indicates that there's also some presence of DMSO2 in plant-grown produce, which means that humans and animals are consuming it as well. And this lack of knowledge then makes it very hard for us to determine or kind of try to predict the impacts that this has on the environment, on agriculture, on human and animal health, all of those things. So all this data points for a need for further research. We need to understand this process more so that we can understand its impact. So I highlighted down here a kind of a motivation, kind of one of the goals for this research. So by gathering kinetic data on this conversion of DMSO2 into inorganic sulfur, we can better understand the impact and the fate of DMSO2 in the environment, which then translates into us better understanding the impacts that it has in human health as well. So this is my current experimental approach. DMSO2, if you're looking at the first picture, it comes from DMS, which comes from DMSP. These are three similar but slightly different compounds. DMSP comes from phytoplankton, phytoplankton that's found near the surface of oceans. They produce DMSP, and then it is turned into DMS, which then kind of evaporates and is released into the atmosphere. And this is actually what you smell when you go to the beach and you smell that smell of the ocean. It's actually DMS that's coming from the ocean that you're smelling. And once it's in the atmosphere, it is oxidized into DMSO2, which is then deposited into terrestrial environments. And in the lab, I will be putting together DMSO2 and the other required reagents. My enzyme will also be added, but the reaction won't actually happen until I add this other compound here. It's another protein called MSUE. This will reduce this SfnG here is called a oxidized flavin, and it needs to be reduced in order for the reaction to happen. And this is what the MSUE is going to do. And once that happens, the reaction will happen. Then after 30 seconds, the reaction will be quenched with HCl to stop the process. Then I will take my products. I will take an aliquot of it, prepare it for HPLC analysis. HPLC stands for high performance liquid chromatography. It is a technique that's mainly used to either separate or identify compounds in a mixture. And the mixture is exactly what I have here. At the end, I also have it in the beginning. So this is a mixture of several compounds, and I'll be using this HPLC device, which is this here, to identify what I have in my mixture at the end, which will then allow me to perform some calculations and identify how fast my reaction happened by identifying how much product I have at the end of my 30 seconds. Then I'll perform some data analysis, and ultimately I'll be calculating these three kinetic parameters here: Km, Kcat, and Vmax. Each of these will tell me something different about the kinetics of my reaction, and then together they tell me how viable it is of a process. This semester, I started working in the lab on the SEMIA building on the 8th floor to actually start collecting some data. So far, I've been purifying the two proteins that I'll need. So SfnG, the main enzyme of my study, which is found here, and then the MSUE protein, which I'll need to reduce this oxidized flavin and actually start the reaction. I've been running some tests. So this is an SDS page that I ran. These are basically multiple steps in this process to purify the protein. And then I took samples from each step. And then at the end, once I had my purified SfnG, these are all for SfnG except for this last column, which is the purified MSUE. I can see that I had my protein in all of these steps. So that's telling me that I do have a lot of purified protein here. I've also been writing my methodology section. As I'm actively working in the lab every week, I'm also writing an actual lab report so that I can report at the end of my study. And in addition to those, I've also started training on how to use the HPLC device at Suffolk. This is the official name here, and it is a newly purchased instrument. It was just bought last year, right before the pandemic. And since the labs haven't been held in person, this instrument has actually not been used yet. So it's really brand new. I'm currently learning how to work it, but once I do, I will actually start running my reaction and monitoring it using this device. The first thing that I'll start doing is doing test trials so that I can determine the ideal concentrations of everything and all of those other conditions that need to be exactly right for the reaction to happen. And once I have all of those, I'll start running my test reactions to actually determine the three kinetic parameters that are going to tell me how viable my reaction is. In conclusion, the main research question of my study is what is the catalytic efficiency of the protein SfnG in the conversion of DMSO2 into methanesulfinate? And just to reiterate, this is this first step in this three-step process to convert DMSO2, this organic sulfur compound, into sulfite, the inorganic sulfur compound. And again, bacteria prefers the inorganic compound because it's much easier for it to get the energy that it needs. Research efforts devoted to this will allow us to better understand the agricultural, environmental, and human health impact of DMSO2 in soil, because again, there is proof that there is bioaccumulation of DMSO2 in plants, which then results in it being present in plant-grown produce, which humans and animals will consume. So there's definitely a lot to consider there, and more efforts should be made to better understand this process. So at the end of my study, I will have determined the three kinetic parameters, Km, Kcat, and Vmax through HPLC analysis. I'd like to thank my research mentor, Dr. Denyce Wicht from the Department of Chemistry and Biochemistry, my McNair Scholars Program Advisor, Dr. Jarvis Chen, as well as the Department of Chemistry and Biochemistry at Suffolk University. So this is the overview of my research project. If you have any questions or would like to learn more about it, please feel free to email me at bsilva2@su.suffolk.edu. And thank you so much for listening in.